about the peripheral nervous system. So this actually covers everything outside the brain. Yeah. Okay, so the brain is under central nervous system, which we'll be covering in another class, and another module totally, another course under Azrina. So Azrina is she's a good person about brain stuff, right? So we've got on peripheral nervous system is everything other than that. So you can see it covers a huge wide range, including the whole part of the spinal cord, right? A little bit about obviously it connects to the brain, but all the spinal related stuff and all the nerves and ganglia which comes out from it, right? So there's the enteric part, which is mainly more specific towards the GI system and all the sensory, etc. as well. Right, so there's again, as mentioned just now, there's loads of references. So for anatomy and physio, because it'll appear in all the courses as the starting of the system. So for example, if you go to third year, you've got anatomy, you've got pharmacotherapy of GI and renal. So before you start the GI, i.e. gastrointestinal module, there will be a part on anatomy and physio as well. So that will be under me, you see me at that time. Right, so yes, you can choose any anatomy and physio books that you like. So these are, I don't think it's the latest version already, because some of the books they update almost every 2 or 3 years. So you can go find out them. Okay, sorry, not in the library because now in the library that day I saw there's lots of new books, lots of them. Right, so class outline. So what we're going to cover in the anatomy and physio is all about the general ones. Right, and bear in mind, before I forget, the presentation that you're going to be doing tomorrow, isn't it? Hey, not tomorrow, next week, for next class, I mean. Those are the main diseases. Why do we pick those diseases? Is because those diseases are the ones that's going to appear in your later classes when we talk about the different drugs, right? So we realized that it could be useful for you to understand a little bit about all the diseases, so that when you go through the mechanisms and so on, when Amy is going to cover, you shouldn't be so lost. So that's why during the next class, although you're waiting for your turn, and also it's good to listen carefully about your other friends' presentation as well, because it will help you in your understanding of your upcoming topics as well. Okay, so my class outline is really straightforward, because a lot of it are the basics that you already learned when you're in your pre-U. That's why it's so straightforward. Like, for example, the neuronal function, right? How does it activate your action potential and all those? Is exactly the same, actually. There's only a little bit of add-on in terms of the details, but most of it is still the same. You still need sodium channels, so it's not like you learn in pre-U is sodium channels, you go to uni is a different channel. No, everything is still the same. So that's why it's not that much of an add-on, actually. So that's why this is the easiest topic among all. So as mentioned just now, anatomically, so you've got your central nervous system and you got your peripheral. So the peripheral is the one we are talking about. So you can see there's also other different parts of it. So as you already learned about afferent, right? So A go into the brain, so E gets out of the brain. So i.e. the signal gets out. So there's the afferent, there's the efferent. So you got your somatic sensory that carries from all the parts that you need to move and so on. So they've got visceral from all the other organs. You got your somatic motor, so that tells you how do you need to move around. You've got your autonomic, so which is like automatic-ish responses. So meaning you don't really have to think consciously that, okay, my heart rate should go up now. Okay, so or things like that. So it's everything is done by, yes, automated or autopilot or autorun. So that's why it's called autonomic. So autonomic, there's two parts. So there's the sympathetic, so whereby you should be alert and all, and parasympathetic, which is more calming. So I hope that in this class you may have a little bit of parasympathetic, because it's supposed to be a story, more like a story, like. So hopefully you can understand it better. So for somatic, is the part that we only know. We don't really go that much in depth of it, right? But it's something that you need to know, which is pretty straightforward anyway. So again, you feel something. So this is the simple experiment that everyone read through it before. So it's your knee jerk, isn't it? So you just tap a certain part under your knee. And then you send the signal, the afferent signal, to the spinal cord. So it passes on the signal very quickly back to produce a kicking. So Just a very simple one. So another example, this is an example of autonomic reflexes. So in this case, the book or the one that I picked, in a way, it's something that is more easy to be sensed by you all in a way, or by us, because it's about your heart rate. So obviously you can sense your heart rate rather than your blood pressure, isn't it? So there's a baroreceptor which actually senses the pressure. So it senses the pressure in the blood, right? So they see that, your pressure goes up when, like, for example, when I talk about exam, and then everybody's pressure, zoop, I have to be alert, right? So, sorry, a bit lame, right? So you send a signal to the central nervous system. So through this glossopharyngeal nerve, so send the signal to the medulla oblongata. So over there, it processes the signal very, very quickly, automatically, and send to the pacemaker. So, okay, we have to re-regulate the blood pressure so that it doesn't go up so much. So for your information, the blood pressure it can be controlled in a very simple way by a heart rate as well. This is one of the control mechanisms. There's other controlling mechanisms like the dilation of the blood vessels and so on, which you're going to go through more detail in English class, right? But for my section, I'm just using it as an easy to understand illustration. So you can actually slow down your heart rate as well, which is pretty quick in rectifying the increase in heart rate. So an increase in blood pressure, because obviously we don't really need that higher blood pressure, right? Like everything else, there's always a homeostasis. So we want something to be in the normal range when the system and everything is in a balance. So the effector organ in this case against the heart, so it again sends to the, what do you call it really? The what's that called? Impulse generator. Sorry, my brain is not. So I haven't been teaching anatomy and physio for heart for some time. So yeah, so it sends the signal to then slows down the heart rate, and then the blood pressure drops, right? Pacemaker cells, yes, pacemaker cells to slow it down. Right, so structure of neuron, again, is something that I already learned. So you can see there's a lot of dendrites. So because of the wide spreading of dendrites, they can connect and talk to different, different neurons at different places. To be frank, can you see my arrow when I point the arrow, or you cannot see the arrow? Can see arrow, cannot see arrow? Yes, no?
Yes.
Yes, can see. Okay, thank you. If not, then maybe I have to change to PowerPoint mode. But I'm sorry, I don't have the PowerPoint for this one. Why? Because for a very lame reason, I don't know why that particular file was corrupted. Thank goodness I still have the PDF. Happened 2-3 years ago for a random reason. So I don't know what's the reason anyway. So the dendrite, so it's widespread, so it can talk to different nerves. So talk to different neurons in a way. So from here, so some of the neurons will actually sort of like be a collector in a way to collect the information to pass on to the next neuron. So you can see there's different collector in a way of information. So to see how great the signal is. So for example, obviously like a simple, I won't say pain is a bit painful. So maybe like a touch signal. So if you are touching, so somebody tap you. So if it's a very light tap, like for example, a mosquito on your I don't like mosquitoes. All right, so on your surface of the skin, so it's a very light tap. So the signal will pass through. So hello, there's something, but it might not be strong enough for the next neuron to collect it. Is that really a signal? So because remember, it's always an all or none response. So if it's a proper tap, like a friend tap on your shoulder, and things like that. So it's a proper tap. So it's a stronger signal stimuli. So it again collect the signal, again causing a stronger signal. So therefore the impulse can jump around. It's the same thing. That's why you got your Schwann cells, and so that your signal can fly around literally, right? Because this part is non-permeable, isn't it? So it can only go through the gaps. So therefore, it can go to the end of the axon terminal, right? So again, these are all same stuff that you've learned before. So for the protection of neurons, just that there's other types of cells around it. So you can see most of them are in the central nervous system, where you got ependymal, you got astrocyte, you got oligodendrocyte, you got microglia. So these are all the brain one. So then this is the peripheral system, nervous system, you got Schwann cells, so the one that protects around for myelination. And you got other, satellite cells. right? So that actually provides all the nutrients as well to support the function of your ganglia, right? So this is how the Schwann cell looks like. So you've got a nuclei and then surrounding it. So again, same information that what you've learned before. Okay, so function again, so you can jump around. So the main signal transmission is ionic. So ionic in this sense, again, is sodium ions, right? So such sodium ions, because it's ionic, can't diffuse across the hydrophobic-ish lipid-based like membrane. So it has to go through an ion channel, right? So the closing and opening of ion channel again will determine whether it can flow through or not. Right, so we've got different types of neurons as well. We've got based on the different shapes of them. So again, this is an overall one. So the sensory are mainly these ones, right, the unipolar. So don't worry, I won't really ask so much about it. You will need to draw about it, so don't worry. Right, so this is some terminology. So again, the impulse passing through is unidirectional. It's not something that can flow up and down. So it's just one direction going. So, but there are conditions such as rabies, for example, it actually causes a retrograde flow. So basically the the virus particles actually flow into the brain from the neurons. So this is where it's something that is not good. So this is just an example of a retrograde flow clinical condition. So there can be movement between the cell body and the synaptic terminals and things like that, right? So retrograde flow is something that is very rarely occurs, and which is not something very physiological anyway. So types of ion channels. So this is something you already learned before. So there's a few types. One is called the leak channel. So this is the leaky ones. So whereby it can open and close just a little bit more randomly in a way. So the more important ones that controls the signal is basically the ligand-gated ones. So basically ligand meaning as what you learned last semester. Hopefully you still remember. So it's something that is like an agonist that actually binds to the active site of your channel, in this case ion channel. So which then causes the channel to be open. So when it opens, so the ions will flow in or out depends on its concentration gradient, right? So obviously the higher concentration site will flow to the lower concentration site, right? You've got mechanical stimulus, so whereby it's more like a touch base. So when you've got a touch-ish sensor, so it sort of like opens up the channel. And there's also changes in membrane potential. So if there's a change, so it sort of switch on the ion channels to open. So there's four main different types of ion channels, which you already learned before. So summary is what you can find here, right? Okay, so it's good to know this. Sometimes when I'm running out of things to set, I'll set questions based on this chart. Very random. Okay, so depolarization versus hyperpolarization. So depolarize is again concepts that you already learned before. So depolarize, so it's what you saw like causing an activation of a generation of action potential. So it's your depolarization. So yep. So examples here is like acetylcholine. So which is which you're going to get very familiar by the end of the next class, by the first class of English one, you see acetylcholine again and again. It's one of your important factors that. So acetylcholine, the short form is ACH, big A, big C, small h. And you've got another very important component called adrenaline, all right, or noradrenaline. There's a few of them. Right, so this one calls the binding. So hyperpolarization is more like causing a switch off. So it's sort of like a minus signal for the action potential. So minus things off. So so there are other agonists in a way like glycine. But bear in mind, hyperpolarization is mainly occurs in the brain in a way. So it's not so much at the peripheral nervous system. So peripheral nervous system is more like an on and off. So obviously the brain functions, the way brain works is a bit more complex, right? So so sometimes for example, there's a pain signal, and then you can sort of, if you hypnotize yourself enough, that you can actually minus off the signal, vice versa. Right, so we've got neurotransmitters and neuromodulators. Sometimes they put different terms about it. So basically, these are the examples. So this is the definition. So compounds that have a direct effect on the membrane potential, or they have indirect effect, or they can have, like gases that can just diffuse through. So again, things you can just get the answers from the textbooks. Okay, so I'll go through the main there's three main components in this class, which is a bit bigger. So it's about the action potential generation. You've got your you got electrical transmission in here. You've got your chemical transmission, and you've got the differences between your sympathetic versus parasympathetic action. So about resting, so you can see here. So you can imagine the whole system is sort of like in a very standby mode, whereby if you need a break, let me know. Okay, if you need a break. So it's in a very standby mode. So because we want things to happen and respond very quickly. We don't want things to imagine like a bee sting you and then you're like, after 2 minutes and you're like, ouch. That is slow-mo, isn't it? Slow-mo cartoon version. So we want things to respond pretty quickly, right? So like a ball is coming towards you, and to quickly avoid it or use your hand to block it off. It's something that you want very quickly. So the whole system is in a very standby mode. So to fit into the standby mode, so there's a lot of adaptations, especially in the chemical ones that we're going to look through later on. So these are the modifications in the way that hopefully we can respond very quickly. So one of the way that responds very quickly, obviously, is the electrical transmission, whereby it's just now you can jump the signal. So everything is resting first. So graded potential. So imagine got different plus signal along. So it's huge enough. So it becomes one signal. Then it can bump, jump around. So until the end of it. So from over here, so there'll be some processing in a way. So to pass the signal to the next one, because our neurons are not so long that it can extend from the leg all the way to your brain. So it's a lot of short ones combining and all the way up. So and all the way down as well. So you have to pass on to your friend as well, i.e. So that's the electrical signal. So this is something that you already know. So I don't know why sometimes I still get very confusing, mixed up answers in exam. Very rare of them, but you still get some of it. So but bear in mind it's the exactly same concept as what you learned in pre-U. So this is resting first. So in resting, what will happen? So the easier way to remember is for so people as not so high capacity memory like me. So I normally remember one version. So i.e. you can remember the activation one or the resting one. So you remember one main one, and then all the other answers will be just up the balik of the other one. So it's upside down in a way. So first thing is your sodium channel. So when it's activation, the sodium ions when the sodium channel opens, your sodium Na+ has to flow from outside to inside, right? That's to activate, right? So outside to inside. So how do you get outside flow to inside flow? So first, during resting membrane potential, you need to pump more sodium. You need to get more sodium outside, right? Is that way? Why terbalik pula? Blur me. So there's a higher concentration of potassium inside, and there's more sodium outside because it's not permeable. So, but however, there's a lot of leaky channels to maintain this, right? So in the leaky channels, there's a lot of more K+ to move around. So the K+ is the one that mainly regulates the whole thing. So K is your... Okay, yeah. So you can see here, eventually, in the end, you will actually get more Na+ outside. So there are more pink outside. Yes, there's more pink outside anyway. Yeah, correct. So there's more sodium outside, so that when it opens, more sodium can go in. So your resting is to make sure that more sodium can be outside. So when it's open, the sodium can go in. So how to maintain this? So we've got leak channels. So the leak channels will allow it to flow around, right? So the details you can read through over here. Right, so only the leak dryness the gated obviously are not open, right? So sometimes it allows it to diffuse around, to move out and in and so on. But eventually the main thing that you want to maintain is sodium more outside and more potassium inside, right? So there's also something called sodium potassium pump anyway to help in the leak channels. So can see when the sodium potassium pump is activated, so the sodium will actually go out. So this is an exchange pump. So the sodium and potassium is at different direction. That's why it's an exchange. So there's some pumps are co-transporter, meaning both go in or both go out. So in this case, one substance go in, like your K go in. Another substance, which is the Na plus, which is more, so it's 3, go out. Okay, so the activation of this pump is actually one of the main one by driving the differences or driving the ionic gradient. Is everybody okay here? It's the same, isn't it? I.e. same like a pre-U, isn't it? So again, sodium potassium pump is an active transport, you need ATP. Okay? If there's any questions, you can ask, no worry. Or everybody tertidur already sampai sini. Oops. Sorry, if in the class I can see your response better. Okay, self-assume lah, huh? Okay, if you can't hear me or putus already, you let me know, if not I can shock sendiri. Right, so this is the part on action potential which you already learned anyway. So we've got this is your resting, right? So those are what happened just now. So if there's a big enough signal, so your action potential will be generated. So your sodium channels will be open. No, so here not yet. So here is resting over here. So you go to the next one. So at depolarization phase, so your action potential will be generated. So you can see your spike here because due to the opening of sodium channels, so the sodium will flow inwards. So therefore the unit become more positive. All right. So all the way the influx of it after it's huge enough. So one signal is generated. So the impulse will pass on to the next one. So therefore, the O part, in a way, you can sort of take a rest. So this is the part where repolarization occur. So during repolarization, the K plus channels open. So remember, there's more K plus inside the cell. So once it's open, so the K plus will flow out of the cells. So therefore, the signal will go down because the charges. So the positive charge between both sides is sort of more neutralized already. It's not so great difference. So it go down until the end. So they have a sort of like meaning resting phase, right? So whereby it's non-sensitive to the new signal, and then you go back to your resting potential again. Okay? Everyone all right, I assume? So in action potential, it's an all or none principle. So there's the mini dip just now over here will be a refractory phase, right? Whereby you can't really receive another signal again. I mean, it can receive, but it won't respond, right? So it's like, i.e., if you have no response now, it's like your brain sort of like jam already. Oops, right, jam already because it's sort of like cannot fit in more information. So based on one of the textbooks, they use the analogy of like flushing a toilet, which is not very, not the best, not the most appetizing one, but it's the same meaning. So either you can flush or you cannot flush. So it is not like in the middle, not much in the middle. So the newer part, which maybe not really that much learned in your pre-U level, is actually this part. So this one onwards is the newer part. So this is where I'll spend more time actually. So the first part here, in a way, that is a bit more new to you, is on the chemical transmission. So this is between the synapse. So obviously the across the synapse, in between the neurons. Okay, synapse is the gap between the neurons, right? So how does one neuron talk to the next neuron? Okay, so how does it happen? So because there's a gap there, so obviously your electrical signal, so your ion channel signal cannot jump across, right? So that's the first thing. It cannot just jump. So it has to change into another form of talking to the next neuron. So i.e., you can have a lot of self-conversation between yourself, but your signal and your friend next to you, it is a different signal. So you actually have to translate your whatever you think inside into words so that your next friend can understand you. So that's the same way. So this is part of your chemical transmission. It's the same analogy. So it's communication between the neurons. So you can't really jump across. So it's mainly chemical. So what are the chemicals that you need? So details, you'll go through TRAS class in terms of the details of the names, because actually a lot of it comes with names. So, but this is the general model. So I'll go through the general model first, And later you let me know whether you can understand. If not, then I'll re-explain this, because you need to understand the general model first. So at the end of the nerves, you can see there's a bigger bulb based on the pictures. If you can still remember the pictures. Where's the picture? Very far in front. So you can see here, there's all these little bulbs, right? So a little bit, i.e. like bengkak, right? Little bulbs at the end, right? So let's go back. So what is in the little bulbs? So the little bulbs here is bengkak, each is bigger in a way because there's actually something storing in there. So there's something called synaptic vesicles. So vesicles in this case is actually like a storeroom. Right, so storeroom for what? So storeroom for neurotransmitters. So remember, it's a whole system that needs to respond very quickly, right? So when you need something to respond very quickly, you cannot say like, so i.e. if, sorry, I don't know why that came across my mind. It's like a bakery shop. So like a bakery shop, you can't just say that, oh, when people want to come in and buy your bread, you can't say, okay, you have to wait for a while. My bread is still my bread is still fermenting. I can't sell it to you. It's still not in the oven. So you can't do that, right? Because it's just too slow. So you want something to be pretty ready, or you can just sell it directly. So it's the same analogy. So you got synaptic vesicles, which actually be a lot of things are sort of like pre-made. So when the signal is here, so it's not it's pre-made. The idea is ready. So while it's resting, actually make all the making process. So actually producing them. So then make it ready and then keep it in the small vesicles. So it has to be in small vesicles, like small storerooms. Therefore, it's not exposed to the outside condition, isn't it? So it's still it's ready made. It's keeping there, but it's not passing on the signal. So when the signal comes in, so it's sort of like so there's receptor binding sites over here as well. So therefore, it sort of triggers the synaptic vesicles, so i.e. the storeroom, or small vesicles to sort of go exocytosis, if you still remember that. So exocytosis, so the membrane will just move and diffuse to this membrane, right? So and then to release the content. So so everything's ready, so you can just release the content. So these neurotransmitters will then diffuse around and bind to the receptors over here, to then pass on the signal, right? So the gap here is really tiny, so you can see the action is, although you have a transition process, but it's still pretty quick for neurotransmitters to sort of diffuse, bind, and pass on the signal. And this response is actually pretty tightly regulated, because you can basically just diffuse across and bind over here in this tiny gap. So remember, there's also other neurons beside them. So there's actually some enzymes over here as well, which actually tries to break down the neurotransmitter, so that the signal, imagine, it won't diffuse across the whole system. Right, then the system will be the signal will be very non-specific, isn't it? But we want a very specific response most of the time. So that's how they regulate it. So it's tight, passed on, and there's also enzymes that helps to degrade at the site. So anything that you want to ask over here? You can on your speaker if there's things that you want to ask. So far, okay? Clear? Or did I lost everyone already? Okay,
clear.
Okay. So just in case the system already put those and I'm just talking to myself. Whoops. Okay, so this is another one. So it's the same thing, but in this case, it's a little bit more words. So hopefully you can, so it's easier for you to do your revision, I hope. So again, your action potential reaches here. So the signal from sodium will be switched into a calcium channel. Okay, so that's change number one. So calcium channel. So calcium channel will actually trigger this. So the diffusion for exocytosis, diffuse across. So again, this one you sort of like trigger the signal for the sodium ion channels to open. So and then it passes on. So where can the drug act? So for example, if there's certain diseases where you want to control the signal, right? For example, signal for we do cover it here, don't we? There's some diseases that maybe causes, like, for example, some drugs that causes like a little bit like paralysis-ish. So you can sort of like, how does it form? So you can sort of block the signal, right? So where can a drug act? So if you want to stop the signal or interrupt the signal, so what can you do? So the easiest way is like what you'll learn in the last semester. There's a lot of agonist and antagonist can. So that's the easiest way. So you've got receptors, yes. So you've got agonist, antagonist. So if you want the signal to be more, you can put more agonist. Got it? So if you want the signal to be cut down, you can just put an antagonist. Cool? So to cut down the signal increase. So that's the most straightforward way, right? So there are other ways, however, which can sort of like taper with the signal. So another more indirect way is, remember the enzyme here when we're talking about. So this enzyme will actually help to break down so that you maintain the signal within around this area, right? So for the enzyme, so you can actually put an enzyme inhibitor. So imagine if you put an enzyme inhibitor, so what happens will be that the enzyme won't work, right? Because there's an inhibitor. So when the enzyme won't work, the enzyme can't really break down your neurotransmitter, right? So most of the time your neurotransmitter is an agonist. So meaning your agonist won't be break down, right? Because if you put in an enzyme inhibitor, because cannot break down. So when it cannot break down, the agonist will stay in the system for a longer time because it cannot be destroyed. So it's a longer time. So i.e. the half, and if you translate into a very scientific term, it will be the half-life of the neurotransmitter will be longer. So when the neurotransmitter is longer, that can exist longer within the synapse. So you can imagine the binding of the neurotransmitter would be longer in a way. It's not like it binds longer to the target. But remember, the binding of the target of the agonist, i.e. the neurotransmitter in this case, it's the endogenous agonist. So the agonist which is within the system, which is your neurotransmitter in this case. It's not a tight binding, isn't it? It's a reversible binding. So meaning it binds, then it activates, then it goes away, and then binds and then goes away. So if the half-life of the agonist, i.e. a neurotransmitter, is longer, so it can bind, go away, so the overall period of activation is longer. So therefore, you can cause an increase in signal. So far, okay? Right, so meaning it's not that it binds longer to one receptor node, because the binding is on, off, on, off. Okay? No response? Should I move on then?
Okay, doctor.
Yeah. Okay. Anyone want to ask can just ask. No worries. It's like in the class. Right. So okay. So again, sorry, I'm putting in a lot of different slides here because these are one of the key concepts that you need to learn for this whole course anyway, because it's tied in from top to toe for the course. So this is again to translate into a more specific term. So hopefully you'll be jelak by the end of the class in a way. So if you're jelak enough in a way, so meaning you already remember them very well. Okay, so for the enzymes and all, so in this case, if we put it into a specific example, I have to give them different names. So this is the system about, remember we talked about acetylcholine from the start. So acetylcholine was ACH. So how does acetylcholine work? So this one, this particular part will be repeated by Ingvi as well, right? So again, just a repetition because it's something which is pretty important to the course. Right, so step one, again, there's a signal coming in. So when the signal comes here, it actually causes the calcium channel to open, so calcium goes in. So you can see here at the background, this is what already happened. So there's the acetyl-CoA and the CoA, and there's CoA which will be turned into the form of acetyl-CoA by mitochondria. So this acetyl-CoA will then be bind to the choline, right? Which is being recycled. So again, another part of our system is that it's very clever enough to do enough recycling, because it just takes too much energy and effort to destroy something so that you can make a new thing all together all over again. So you can actually bind to the choline and then store it in the synaptic vesicle. Then it can be released. So after binding, so there's the enzyme here that breaks down, isn't it? So in this case, for acetylcholine, the enzyme that breaks them down is called acetylcholinesterase. So remember chemical terms. This is an ester bond. So you can put in So there's an esterase enzyme. So to then break down into acetate and choline. Yeah. Okay. So the choline is the part that can be recycled and goes back into the synaptic cleft again. It's not into the presynaptic neuron. Is that okay? Excuse me. Right? So this is the synaptic cleft, sorry. The cleft or the synapse is the gap between that. So far, okay? So, i.e., if you're finding yourself a bit blur and whatsoever, just refer back to the diagrams again. Then you get your answers. Okay? Right. sorry. Okay, so this is another one. Sorry, I get a cough. Right, so there's an inhibitory. So there's parts whereby, remember the signals, they can talk to each other in a way that there's a different gathering of different information. So, but bear in mind, again, this will be more towards a central nervous system, but I thought of just showing you anyway. But again, I won't be asking your exams, just FYI. So there can be inhibition at the presynaptic site. And also, or there can be excitation or more. So what happened in this inhibitory? So whereby there's other neurotransmitters which are more on the neurotransmitters that we mainly talk about in the peripheral nervous system are the positive ones. So meaning when I have more of it, I'll turn on the signal. This is what it means by positive ones. So there's others that are negative ones. So for example, like GABA. GABA is a neurotransmitter but when it's activation, it actually causes a negative signal. Because, for example, the GABA, it binds to the calcium channel. It actually causes lesser calcium to enter. So this is like a negative signal. But this one, it can act on the presynaptic, the pre-neuron, right? So there's others, for example, over here, which is this is a positive one. This is another positive one. So this is called serotonin. So again, it's more in the brain. Both of it is found in the brain. So the serotonin, so it can actually bind again to activate the calcium channels. So this is a positive signal. So imagine if your action potential here is maybe like, okay, this is a signal they need to pass on. So with this, so it causes more. So the signal is augmented. So for the GABA, the signal is minus off, right? So you can see that it's so here is like a gathering of information in a way, in a very short term description. So they sort of gather. So based on the talk between the different neurons, so what's the final result? I pass it to the next person after the discussion in a way. So here, there's another part. There's also modulation at the postsynaptic site. So just now it's about the presynaptic site that you can change. So there's also postsynaptic changes in the way that it can happen. So this is a different term of saying it. So there's something called summation. So summing, sum up. So it's like a add on. So you can see the signal, sometimes it comes one by one. So then you get a bigger signal in total. So sometimes it's two comes at the same time. So you get one signal in total. So it can be this way as well. So here I'm just trying to jump a little bit off topic. It's part of the autonomic nervous system. You have sympathetic, you have parasympathetic nervous system innervation. This is something very, very, very specific to the GI tract. So we've got a different name for the whole system called enteric nervous system. Okay, so this is just for GI tract. Okay, so you can see that in your GI tract, in the muscles, so obviously there's different layers and so on. So the enteric nervous system is mainly over here, right? So what happens is that, so it can be myenteric nerve plexus, submucosal nerve plexus. So there's a neuronal control over here as well. So in a very simple terms, like for example, sometimes if you're very nervous, you might find that some people say, I need to go to the toilet again and again. So some people is like, oh, I feel like I need to toilet, i.e. I get diarrhea a little bit, like when I'm very, very nervous. So how does it happen? So this is how it happens. It goes through this enteric nervous system way. So to get to that your signal sort of passes on, and it sort of like causes more peristalsis. So this is how it works. So, but remember, in the gut, there's also other system which is involved. So there's also the hormonal system which controls. So the hormonal system is the one you can sort of ignore it, the details at the moment, because this is, and the local factors. So this is the part that you're going to cover more in the GI system of the anatomy and physiology. So more for you is just this neuronal control. Excuse me. So you can see there's short reflexes, right? So there's also the long one, like myenteric plexus, like the one that mediates it. So you got stretch receptors, you got chemoreceptors. So again, depends on what you eat, right? Chemo, so it's like the chemicals in the food that you're getting, and you got stretch, so how much you're taking it as well. So how much you can stretch around, because based on the amount of food or fluid that's in your body. Okay, so that's in your GI tract, not really in your body, right? So you can see, so there's different that control the waves and so on. So you can see these are all the terms in a way that you can see in your autonomic nervous system as well. So this one just bear in mind as well. So yeah, some of the things that I can set when I'm running out of things to set. Okay, differences between the system. So you got somatic and autonomic nervous system. So there's a comparison there. There's also another comparison between sympathetic and parasympathetic nervous system. So this is a class whereby I sort of like spot the difference. So first, so on the one side, so now somatic, another side autonomic. So this side is autonomic, this side is somatic. So what's the difference? So okay, I'll give you a minute or so to look at it. So then spot the differences yourself. So hopefully you find a few differences. Okay, back. So you can see here the system, okay, autonomic pathway. So you can see again, of course, there's a sensory one. It passes signal on. So you've got an extra here, isn't it? Autonomic ganglion, right? And you've got everything else is about the same, right? This part is all the same. You go through the interneurons and the spinal cord and so on. That's the main difference. So because of this difference, you can actually have it's split up into two different neurons, isn't it? So you got preganglionic axon, you got a postganglionic axon. Okay, right. So so therefore, them up into different ones. There's other differences, of course. You've got voluntary, some are involuntary actions, right? So neuronal pathway, so obviously autonomic. Remember, there's a pre, there's a post, right? So they get sort of like gather the information and then have another one that goes to the end, right? So you've got differences in your neurotransmitter. So bear in mind if it's somatic, so it's more like a movement muscular basis ones, because remember your effector, your target effector, i.e. your target organ, is on the muscles, right? So this part, if autonomic, is basically everything else. Yeah, literally everything else. So in skeletal muscles, so you can see a neurotransmitter is just ACH. But autonomic, you have ACH or norepinephrine. So again, American and British, they don't talk very well sometimes in terms. So norepinephrine or noradrenaline is the same thing. Adrenaline or epinephrine is the same. Okay, so so these are the terms. So these are the differences. So another one about sympathetic and parasympathetic. So it's something very simple as well. So imagine, so sympathetic is your fight or flight response. So you're like very nervous. So people give a lot of textbooks says that it's like you see suddenly a bear chasing you and all that kind of stuff. Tiger chase you. those examples that we've been using in the textbooks. So me is more like, why don't you maybe exams. I think that's good enough for sympathetic. So if maybe it's a driving exam or whatever exam you're talking about. Or sometimes even a presentation that you need to go up the stage, like stage. So that'll be a sympathetic. So obviously to to take into account of all this. First thing, you need to be increased in alertness, right? So all this helps in all sorts of things. So bear in mind, so here a lot of things are increases, dilates. So some are inhibition, some has no effect, right, on the bladder. So parasympathetic is the calming one. So imagine everything is done, and you're at the beach lying down, there's no worries, there's no exam, no work, no assignments, happy good day for a while. So that's parasympathetic. Okay, so in short terms, you say you're resting and digesting state, right? So again, spot the difference. Sympathetic versus parasympathetic. Okay, so I'll give you a minute again. So spot the difference. So look at the diagram and see what you can find in terms of the differences. Sorry, I just get a drink. Okay, cool. Can you spot the difference now? Right, so sympathetic, I don't know why they use blue. Parasympathetic, so you can see here. So first thing, do they go to all the organs? I think the answer is yes-ish. Yes, it goes through everything. So sympathetic is the one that have something extra. Right, so they call it sympathetic chain. Right. Here you have, so you can see here it passes through here and then goes on. Right, so they guess this is the pre, this is the post. So pre, post, pre, post. So you can see here, not all the neurons in a way, not all the organs, sorry. Excuse me, sympathetic will go more, it goes to the adrenal gland, but obviously parasympathetic, it also got. Not directly towards the adrenal gland, right? Right, so so you can see this one has a very long pre. Okay, some they got mini post, so you can see there's still an intersection, but intersection is very short, small, and tiny. You can see it. Right, there's the postganglionic, but this one is a long postganglionic. Right, and yeah, it goes through everywhere. Anything else? So you can see some, if you want the details, it goes through the middle part here for the starting. So here it goes through the top, different sections from CN 3, 7, right now. And go through here as well to go out to signal. So the others in between is mainly from sympathetic. Excuse me. Anything else? So I here is medulla oblongata. Here is spinal cord. Bum, 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 bum. So I think that's it mainly. Okay, so details will be over here. So these are just translating into words, right? So you can see there's multiple over here, right? Sorry. Long time didn't speak for so long. Right, so there's multiple over here. So here, sympathetic, bear in mind, the main neurotransmitter that we're talking about is adrenaline and noradrenaline. Right? There's not much of the acetylcholine system. Okay. So most of it is adrenergic, right? So very rarely there's ACH, very, very rarely, right? So these are the main ones. So we mainly talk about the main ones. So to reintroduce you some of the names. Yes, I'm all right. Thank you for asking. That's why I'm actually working from home a few days because long story. I want to tell after I switch it off the recording. And I'll stay there forever. Goodness. Okay, so so there's this adrenergic that secretes NE. So the adrenergic system actually has the adrenergic receptors. Okay, so there's alpha and beta adrenergic receptors. So they got the alpha and the beta. So again, terms that You would know more their function and so on later on in AB's class. Parasympathetic. So as mentioned just now, it's mainly from the medulla oblongata and a little bit of the pelvic nerves. So it's sambong. So you can see the post one is really short, is within the organ, right? So most of it, or all of it, is under ACH. So ACH mainly binds to the nicotinic receptors. Okay, so i.e. nicotine. So the easy way to remember is your cigarettes, kan? Smoking cigarettes. So that's the nicotinic receptors. Okay, so nicotinic receptors, and you also got a group called muscarinic receptors, right? So again, terms that you hear more at the end. Okay, so these are again comparisons. So to put into words, left and right, what's the difference? So again, something that you should learn and you should know anyway. So you can see this again to put into terms. So sympathetic, so it's this way. So you've got something bigger in between. So you can NE, NE. All right. So parasympathetic, again, it can be, so it's more ACH, ACH. So sorry, sympathetic here is ACH, but the last one is NE. So, but in terms of effects, we mainly look at the last ones, right? Because the last ones are the ones that you sort of, how does the organ respond? That's this one. Okay. So I think that's again what are the what's the different response for different organs. So again, you can just go through them. So again, these are very famous exam questions. So again, if I want to set scenario, so these are examples of the scenario. So this one, how does it happen? So do I have? No, I don't have the answer as well. So you can find out if you're interested. So this is just an example of scenario-based questions in a way. Sympathetic chain located. Sorry, I'm just looking through the questions. Where is the sympathetic chain located? Good question. I think it's here. This one. Yep, this one. Sympathetic chain. Where is it located? Good question. I never really checked. Can I get back to you? Or maybe I just do a quick Google now. No, it's still within the nervous system base. Wait, sympathetic chain location. So external to the spinal column. So wait, let me go through the images so maybe I can show easier. So basically it's beside the spinal cord. Beside the spinal cord. So you can see here. Wait, let me change my share screen. Is that okay? Because I think this one almost finished anyway. Wait, where am I? Here. Okay. Stop sharing. Oops. Okay, share again. Share content. Wait. What in my Safari, where is it gone? Share the whole screen. So can I? Okay, share the whole screen. Okay, so yeah, if you find Google very quickly about sympathetic chain, so basically the easiest diagram is this one. So it's something directly beside your spinal cord. Okay, something beside. So there's an external structure just beside it, running along with it. Okay, so this is the clearest lah. Okay, since you are share full screen, also okay. Wait, sorry, I just go to the bottom. Actually, it's here, ma. Wait, okay. Okay, so that's it. So a little bit about peripheral nervous system repair and diseases. Is it clear if I put it this way, or you prefer me to just share the Acrobat Reader per se? Would it be clearer that way? Any response? No. Okay, I think I'll just stop this and I go back to Acrobat then. Both can. Okay, never mind, I stopped already. Oh no. Damn weird. Wait, you don't know lah. Never mind, I'll share desktop lah. I don't know why the option for Acrobat Reader is missing now. Oh, also a lot of weird things. Well, we can just go through them. Okay, so injury about peripheral nervous system. Okay, this is a headache. So literally any type of injury to the nerves is a big headache. Why? Because as you all know that the nerves can still grow when we are really young, like when we are babies and kids, like really young babies and all. They grow, right? The rate of growth of neurons is much faster. After we are adults, or I think big enough kids, I think our repair rate is it's just so slow. It's like millimeter rate per, I don't know how, per day or per hour. It was crazily slow. Crazy slow. So it takes years, months to repair it properly. So yes, so this part after its injury, so you can, the whole thing obviously get injured, right? So the whole thing's all like fragmented or kaput already lah, literally. So your macrophage will come and gobble up all the rubbish because it's already rubbish because it's corrupted. So to clear the system and then to build a new one. So this is the one which is difficult. So then, yes, there are nerve growth factors and all which is secreted in the system. So slowly it can grow and go back to normal. Right, but it's a very slow and painful process. Not painful in a way pain, but it's a painfully long process. It's just so, i.e. in the middle, you have to be very, very patient because there's not much changes literally. Okay, so if we go on, another one is about shingles. So later I'll switch off the recording, then we go more story time lah. Right. So some stories I may not want to disclose so much. So shingles in a way, so it's another one. So it's basically by the chicken pox one. So it's something that last time when I studied, I don't really study that much about it. But this is something that you should know more in a way because it seems to be getting more popular lately. More popular. There's more people around me that gonna. Okay, so it's a disease whereby it's literally it's the chickenpox virus. It's exactly the same virus. So therefore, they say that if you're gonna chickenpox one, so there can be some dormant virus in around, and if you have low immunity and you're stressed, etc., I don't know what on earth is the trigger anyway. You might trigger it. So don't know. It depends on which nerve it affects. So then you can cause your the symptoms. So the symptoms, in a way, it's the first thing is painful. It's a bit of rash, so it's not very clear sometimes. So it goes around. It can goes around body. The easier way, normally it goes around like a horizontal way. So in this case, because it follows the nerves, this literally follows the nerves and goes around. Okay, so there's other diseases as well which are not so popular, but you can still see them here. Okay, so diphtheria is something that again it's in your vaccine already anyway. Okay, so that's the end for the class for this one. So I'm just trying to show you the second part here. So I actually share another one which is add-on file. So here just a little bit of add-on because apparently it's I won't really ask you like in a way like which one is C2, C3, which is D2. So these are the terminologies that you use. So this is cervical plexus C1 to C5. These are more towards medical, isn't it? So you've got your thoracic nerves. So you got your T's, right? And you've got your lumbar nerves. So you got your L's, and you got your sacrum one, so which is your S. So you can see these are all the different numbers. So again, as I say, I won't really ask you which is what and all because it's not really very pharmacy. So it's mainly the medical, but it's still something you need to know roughly what it's about because it's in case when you go to clerk your cases in the future in the hospital, especially in the neuronal ward. So sometimes they got a damage and so on in which section. So it will be stated in your notes. So maybe you say which part of the T. So for example, if it's your some people that have back problems, some you've got your what do you call it, the disc, not dislocated, but the disc, which is, what do you call that? Slip disc, for example, and things like that. Which part of the lumbar thoracic canal? So these are the things that you will see in the notes. So just in case it doesn't sound too weird or to you. So these are the different parts of the spinal cord segment. So anatomically, right, you can read through them. So there's another one about the sensory output. So this one, again, you can go through because I think I go through a lot of it already. So this is the autonomic one. So this is the skeletal one, right? How it works. So, so the injury. So again, which part of the injury? So if the injury is at the bottom, it's not so. So it mainly affects the legs because this is where the nerves output to the legs. So if you go further up, damage, so the more dangerous things could be and so on. Okay. So that's it. That's it for the class per se. Any questions you want to ask? Right? Anything you want to ask?